Hello everyone, let us get started. We have waited enough for more people to join. I will be sharing my screen. Let me know if the PPT is visible. Hmm. What about fine. It will be great if we can see some more faces. It looks better on the YouTube video after this recording goes online. Okay, to start off, let me welcome you all to the first week of the PMRF-NPTEL lecture series on molecular biology. This series, as you would know, is where we try to solve different problems from the previous year's run of the course, and we will go through questions and learn how to solve them various questions so that we get a better understanding of how to solve get a better understanding of the entire course as well as how to tackle these questions in our exams as well as the quizzes. I will briefly introduce myself. My name is Anuvab Chatterjee. I'm a PMRF fellow at IIT Kanpur, and I will be taking this lecture series. And today we are going to discuss about the basics of cell biology, how the prokaryotic, the eukaryotic cells look like, what are the different organelles, essentially the basics of the basics, so that later on we can actually move on to hardcore molecular biology, where we'll learn how to manipulate all of this in order to use in various applications. So first off, I would like to know if anyone has any questions from this week's lectures, the lectures 1 to 5 that were released. If anyone has anything, then we can tackle that first. Otherwise, we'll go right into the PPT. Okay, so I guess we go ahead, and how this is going to go is that I'm going to show you a question and I'm going to give you a bit of time to choose one of the correct answers, and then we discuss the concepts behind the questions, and then we finally answer. So we learn as we go along. So the first question that we have is sugar chains in peptidoglycan layer are composed of which amino acids? So the peptidoglycan layer, as you would know, is part of the cell wall of prokaryotic bacteria. And the question asks us which amino acids actually compose this peptidoglycan layer. The options are aspartic acid, L-aspartic acid, D-glutamine, L-lysine, and D-serine, L-alanine, D-glutamic acid, L-lysine, D-alanine, L-glutamic acid, D-phenylalanine, L-threonine, D-valine, L-proline, D-tyrosine, L-glutamic acid, and D-glycine. So we have a lot of answers coming through. Let me just turn the in-call messages on. Uh-huh. Okay, so everyone has answered B, that it is L-aniline, D-glutamic acid, L-lysine, and D-alanine. Okay, Integrity Innovator has answered A, which is L-aspartic acid, D-glutamine, L-lysine, and D-serine. So let us quickly look into it. So you would have seen this slide during the presentation as well. So what is going on here? So prokaryotic cells have a thick cell wall outside the cell membrane, and this cell wall is made majorly of a layer of lipids and sugars, which is known as the peptidoglycan layer. So the peptidoglycan layer has a lot of sugars which are called NAG, which is N-acetyl glucosamine, and NAM, N-acetyl muramic glucosamine, which is attached in horizontal chains. So I will move this to here, and you can see that these horizontal blocks are NAM and NAC blocks that are attached to each other, and in between vertically these are connected by a lot of peptide chains. So peptide chains go in between these NAM and NAG blocks vertically to connect them together into a solid cell wall. And these amino acids are, as everyone has already suggested, L-aniline, D-glutamic acid, L-lysine, and D-alanine. So essentially, we have a composition of different sugars which are attached to one another, and again another cross-linking of different peptides to join them together to form a solid cell wall. So the question is, do all prokaryotic cells have the same cell wall? The answer is no. There are actually two types of prokaryotic cells, which can be classified as gram-negative and gram-positive. We'll get into that in a bit, but essentially, so one kind of bacterial cell has a thicker cell wall of peptidoglycan, whereas the other does not. It has a thinner cell wall and also has a membrane, or also has a layer on top of it. So let us look into it. So two types of bacterial cells, they are gram-positive and gram-negative. The gram-positive bacteria, they have a huge thick cell wall made out of a huge chunk of this peptidoglycan. So the NAM and NAG blocks as well as the peptide linkages. So you can see that a huge layer of peptidoglycan covers these gram-positive bacteria. Whereas on the other hand, gram-negative bacteria, they have a very thin peptidoglycan layer, as we can see right here. So compare this to this right here. And on top of this peptidoglycan layer, they have another layer of lipopolysaccharides that cover them. So gram-negative bacteria have a thinner layer, gram-positive bacteria have a thicker peptidoglycan layer. And why do we call this gram-positive and gram-negative? This is because of a particular staining that has been done historically throughout microbiology to separate these two kind of bacteria. So we'll go and look at that once. So this is known as gram staining. So what gram staining does is it tries to differentiate between this kind of bacteria, gram-positive bacteria with a thicker peptidoglycan layer versus the gram-negative bacteria with a thinner peptidoglycan layer. So how do we do it? So first we take our bacterial cells, we fix them on a plate, followed by our primary staining technique, which is known as primary stain, which is known as crystal violet, which colors all of the bacteria purple in nature. So you can see they all of them have turned purple or violet. Then we put in iodine, which fixes the crystal violet into the cell wall as a huge complex, which is difficult to wash out. The next step is decolorization or alcohol wash. What the alcohol wash does is that it takes off the top layer of the cell wall. Now, if you remember, gram-positive bacteria have a very thick cell wall, so they do not let go of the crystal violet and are still purple colored after the decolorization. But gram-negative bacteria have a very thin peptidoglycan layer. All of it essentially gets washed off and they become colorless. And finally, you then counter stain with safranino, which is a red stain. So only the gram-negative bacteria are in the position to take up the safranino and then turn pink or reddish in color, whereas the gram-positive stay the purple because they never let go of the crystal violet and iodine. So this is a very interesting technique that was used to initially classify bacteria, even when we didn't have much knowledge of what exactly makes up all of these layers. Just depending on the cell wall thickness and the properties of the cell wall, we can classify bacteria, and this technique is still regularly used in microbiology labs. So gram-negative and gram-positive bacteria. And coming back to sugar chains in peptidoglycan layer, and we have already talked about it, it is B, L-aniline, D-glutamic acid, L-lysine, and D-alanine that connect the different NAB and NAG groups into one thick peptidoglycan layer in prokaryotic bacteria. Okay, does anyone have any questions from this? Anything to ask about from the gram staining? So the best way to we deal with these sessions is that if you have any questions, then you ask, so that that is the way we get the most out of these sessions, because these are designed in order to bridge the gap between the NPTEL lectures, which are just videos, and the students who have questions. So Aratrika asks, so the role of iodine is good point, Aratrika. So crystal violet by itself is a dye that can suffuse into the cell wall. What iodine does is that it makes a complex with crystal violet and causes it to be difficult to be washed off. And that is why during the decolorization step, the gram-positive bacteria do not let go of the crystal violet. Otherwise, the alcohol would still have washed off the stain. Iodine causes it to get fixed on top of the cell wall of the gram-positive bacteria. Is that fine? Okay. If you have no further questions, we move on. The next question. Blank is responsible for spread of antibiotic resistance across the bacterial population. The options are ribosome, flagellum, pili, and plasmid. Okay, I will give you a second to figure this out. Okay, so let me look at it, and most of us have answered it as D, plasmid. Okay, so let us look at it. So what exactly is antibiotic resistance and what exactly are all of this? So plasmids, so the answer is going to be plasmid as most of you has pointed out. So what exactly is a plasmid? So in case of a prokaryotic cell, so in case of prokaryotic cell, which means bacteria, the DNA, genomic DNA, which actually codes for majority of the genes are not present in the nucleus. It is present in the cytoplasm along with everything else, and it looks something like this. So a jumble up of chromosomes, or a chromosome rather, a circular chromosome that is attached to the cell wall and just floating around in the cytoplasm. But in case of bacteria, there are also some extra chromosomal DNA fragments. So these are known as plasmids. So what are plasmids? Plasmids are circular DNA which are present in the cytosol of the bacteria that can by itself undergo replication, transcription and translation, as well as code for various genes that may help in survival of the bacteria overall. And the interesting thing about these plasmids are that, as I said, they are independently replicating as well as transcribing using the machinery of the bacteria itself, but also can be transferred between one bacteria to the other. So if one bacteria has a very interesting plasmid, it can just pass it on to another bacteria using something known as conjunction. So this is extra. So the points are this extra chromosomal DNA, not part of the chromosome. This is circular in nature normally. This is independently replicating, so it can replicate by itself. It has an origin of replication, and it can be passed on from one bacteria to the other. So why is it important to know about plasmids? So we will get to know uh, it here. So this is a map of what a plasmid looks like. So this is our in this diagram, this is our circular genomic DNA of the bacteria, and this is our plasmid, and this is a blown up picture of the plasmid. So as I said, the plasmid has the origin of replication, which means by it is independently replicating and can make multiple copies, as well as has something called a antibiotic resistant gene. So what is an antibiotic resistance gene? So we know what antibiotics are. These are chemicals or different compounds that we use to eliminate or kill bacteria. And this can, like the most famous all of us would know, is going to be penicillin. So what antibiotic resistance genes do is that they confer the bacteria resistance to the antibiotic. So even in the presence of the antibiotic, they can survive, they are not harmed. And they essentially can grow even in the presence of the particular antibiotic. So this is really important in something that has been growing up very recently, which is known as multi-drug resistant or drug resistant bacteria. You would know that before COVID, there was this huge talk of virus known as it was MI multi drug resistant staphylococcus, which was like a huge plague throughout Africa that was spreading around. So this particular bacteria had gained resistance to multiple different antibiotics because it had plasmids from everywhere else. So it so essentially, if a bacteria has a plasmid that confers antibiotic resistance, it can get it is not affected by the antibiotics we use to treat it normally. So, and this has become a problem as more and more drug-resistant strains have been coming forward, which are resistant to particular antibiotic effects. Further, plasmids are also really interesting for us molecular biologists because plasmids, as we have said, are independent, and anything you put into a bacteria, into a plasmid, can by itself replicate further and further. Say I have an interesting gene such as insulin. If I put it onto a plasmid, then more and more of the gene will be produced by the bacteria, and then I will have a lot of insulin on my hands out of nowhere. So this is how recombinant insulin that you probably would have heard that people with the diabetes use a lot of insulin. All of these are actually made using these plasmids and bacteria. So we put in insulin gene into the plasmid, and we grow bacteria, and the bacteria produces a lot of insulin for us. We harvest the insulin, and then we package it and use it to treat diabetes. And we have used it for a lot of other things as well, but we will get to know more about how we use plasmids later on in this course. And finally, the structure of different forms of plasmids. So even though we almost always draw plasmids circularly, as we saw in the previous example, this is essentially how we draw them, or how they should exist. But in the physiological conditions, essentially inside the cell, they do not exist as circular in nature, because that is very difficult to maintain a perfect circle inside physiological conditions. What they do is that they undergo supercoiling, which is something like you would have remembered how the landline phones used to exist in way past when, where the landline phones had these cords that turned amongst itself and formed the spirally structures. So that is how a supercoiled DNA is formed. So a circular plasmid undergoes normal reconstruction to form a supercoiled DNA, which is much more stable, and it stays supercoiled normally in the cell. So one type of plasmid that you can see in the cell is called a covalently closed circular plasmid, which is CCC, or essentially a circular plasmid, or a supercoiled plasmid which just coils amongst itself and forms a very densely packed plasmid structure. Or you can have an open circular DNA, or a nicked plasmid, where by some event or the other, one of the strands of the DNA, obviously you would know that plasmids are double-stranded circular structures, they are not single-stranded DNA, one of the strands have broken. So they have a nick. It is still circular, but it is nicked at one end, and because of the nicking, the supercoiling sometimes comes off. So these three kinds of structures are what we majorly see when we run a plasmid on a gel, or like look at a plasmid. So you can expect all three of these. So yeah, we have a question. Let me look at it. How do the plasmids achieve supercoiled structure? By loosely turning. So plasmids generally do. So have we? If you have ever seen a rubber band, which is closed circular, and if you just keep on turning the two ends, it just keeps on coiling amongst itself. Plasmids do something like that. In order to prevent its, so I would like you to revisit the organic biochemistry that you probably would have taken before this course. If you had not, that is not a problem. We'll probably deal with it when we get along the course. So DNA structure has essentially two parts: phosphate backbone, which is polar, and which isn't so it is a phosphate backbone which is polar, which tries to always face towards the outside of the plasmid, because outside the plasmid in the cell it is always full of water, so it does not the polar part likes water, but inside to the DNA are nitrogenous bases which are hydrophobic in nature. They do not like water, so the more it coils, the more it tries to hide the organic and the hydrophobic part inside itself. So it tries to turn, but there are other enzymes as well, such as topoisomerases, which help in this negative coiling as well. So negative supercoiling increases, and then they can form a very compact structure. So that is what generally normally happens. So this also happens with our DNA as well. You will see, you have probably seen during early biology classes that DNA before replication occurs is formed into compact chromatin structures. That is all because open structure DNA is very unstable because the organic groups might face the hydrophilic environment and then break down. So they always try to hide it and then try to turn amongst itself to form a ball-like structure, which is the supercoiled DNA. How the plasmid achieves supercoiled structure by losing turns. Hmm. So if you again coming back to the rubber band image, if you ever have taken two ends of a rubber band which is circular and kept on turning it, it would supercoil. It would form a compact structure which is turned in on itself. But the moment you let it go, it just bounces back into a normal circular rubber band. That is because all that strain does not have any place to go. That so in order for the plasmid to stay supercoiled, there are these enzymes called topoisomerases. What they do is that after the plasmid has turned into a supercoil, these topoisomerases go and sit on the plasmid and cut one of the strands and then join them essentially in this eight kind of a formation. You can see right here. So instead of a circular one, you cut one of the strands, one of them go here, the other goes behind it and forms a eight kind of a structure. When you do this cutting and pasting, the strain actually gets released. So it can stay in a supercoiled structure, but the moment you let it go, it won't bounce back to a circular form. So this is how topoisomerases help in supercoiling of the plasmid DNA. Is that fine? Okay. And Aratrika has asked, isn't that gaining turns? I do. Maybe I answered it with the topoisomerase explanation, but if you have anything else, Aratrika, you can ask again if you want to know anything more. Yes, the figure does show the topoisomerases turn supercoiled into circular. So there are different types of topoisomerases. There are type 1 to 4. Some of them they can go from left to right, some of them can go from right to left. But there is also, yes, this is an enzyme, again another enzyme present, which is known as DNA gyrase, which also helps in all these supercoiling things. But topoisomerases can do it either or. Topoisomerases are just enzymes that can cut and then rejoin DNA. But if it is helps in positive or negative supercoiling, that depends on the type of topoisomerase that is present. So they are like 1 to 4, and depending on which one of them can go from supercoiled to normal, and the other can go from normal to supercoiled. Actually, so in the PPT, it is said the circular form becomes supercoiled by losing turns. So the more turns would lead to more supercoiling. So a bit of confusion. Uh, So in the PPT itself, the interchange between the different forms are possible under the in vitro and vivo condition, such as DNA gyrase, which produces additional turn into the super circular DNA to adopt supercoiled formation. So additional turns allows it to form supercoiled. So now there is also this question of which side you are turning. So supercoiled DNA exists as a negatively supercoiled structure, which is counterclockwise rotation. It always wants to turn counterclockwise. They never exist clockwise turning because the shape of the DNA, like there is steric inhibition due to the how the phosphate backbones are arranged. So uncoiling or losing turns can either mean you go from negative to zero or zero to positive. But essentially, whenever someone is talking about supercoiling, more turns means more supercoiled, less turns mean less supercoiled. Circular form becomes supercoiled. Okay, perfect. So blank is responsible for the spread of antibiotic resistance across bacterial population. The answer is obviously plasmid. Perfect. Moving on, chemotaxis is A. Movement of bacteria towards a chemical gradient, movement of bacteria away from a light source, movement of bacteria in response to temperature changes, movement of bacteria towards oxygen levels. So we have a string of answers just still coming through. So I'll give you a second to try and answer this. Okay, so everyone has answered A. We are still having more answers coming in as A. So just let us look at it. So chemotaxis can be divided into two separate words. Chemo and taxis. Chemo meaning something to do with chemicals and taxis meaning movement. So chemotaxis can represents the movement of bacteria in response to a chemical gradient. It can be of two types. One is a chemoattractant gradient, so bacteria sees something it likes, such as sugar, glucose, some place to nest, some anything, and it tries to move towards the source. So, say the bacteria moves to the source of sucrose, and the bacteria uses flagellum to tumble and roll towards the chemoattractant. It can be the opposite thing as well, which is a chemorepellent. Says the bacteria senses something toxic, such as an antibiotic, which is it is not resistant to, or heat, not heat, like a chemical resource which it does not like. So in that case, it will move away from the chemical gradient. So that is called chemorepulsion. So chemoattraction and chemorepulsion, both of them together, call them chemotaxis. So that was pretty straightforward, and everyone has also answered it correctly. Chemotaxis is movement of bacteria towards a chemical gradient, like movement. Even though the question says towards a chemical gradient, it could be away as well. I would rather pose it as movement of bacteria in response to a chemical gradient. Any question from this one? Okay. Ha. So we go into plasmid isolation. So as I had said previously also, plasmids we have essentially turned plasmids into our own plasmids and then in turn bacteria into our own factories to create more and more DNA, proteins, a lot of other things for us. Insulin being a prime example in which we have created recombinant insulin, which lets us harvest the insulin completely from the bacteria. And this we can use to but this asks the question: how do we isolate the plasmid in order to use it? So this the most commonly used technique in plasmid laboratories are plasmid isolation using alkaline lysis. And the question here asks, in plasmid isolation, the purpose of alkaline lysis is the options: to enhance the binding of plasmid DNA to a spin column, to denature chromosomal DNA while keeping plasmid DNA intact, to selectively denature chromosomal DNA and plasmid DNA, to digest RNA molecules. Okay, so I'll give you a moment to figure this one out. So what exactly does alkaline lysis of uh, alkaline lysis based plasmid isolation do out of these four? Okay, so we have a lot of answers again. So we have some C's, which is to selectively denature chromosomal and plasmid DNA, and then we have a B to denature chromosomal DNA while keeping plasmid DNA intact, and then a mix of C's and B's. Okay, so let us look at it. So isolation of plasmids using alkaline lysis method. So even though we have just touched upon it, this actually is a bit complicated. So I will try to go through it slow so that we get an idea of what is happening. Okay, so we have more and more answers coming. Okay, so the alkaline lysis method. So first thing is that why is it alkaline lysis? Why do you not use acidic lysis to lyse the cells? Can anyone tell me? I don't know if you guys have ever done a molecular biology course before this, but I think does anyone want to have a guess? Why is it called alkaline lysis and not acidic lysis? Yeah, that is pretty much on point because DNA is already acidic, but then why alkaline lysis? What would that help in? Like you're almost there. You just have to complete the sentence, Anatrika. DNA is already acidic, and we use alkaline lysis because to neutralize and denature it. Not really. Uh, so that it comes, so we can dissolve the DNA. So since it is acidic, you can use an alkaline solution to put it into solution. Acidic things do not dissolve in acidic things. Acidic things dissolve in alkaline things. That is why I use alkaline lysis method to dissolve the DNA and get it into the solution in the first step of alkaline lysis. Yep. Okay. So what do you do first? You first grow your bacteria. So you take a conical flask, test tube, whatever, you grow the bacteria and get a lot of bacteria. And then you do something called centrifugation. Centrifugation is just spinning the test tube at a very high rate so that most of your bacteria pellets down as a thick white pellet at the bottom of the test tube. Then there are three different steps to the alkaline lysis method. So when you pellet the bacteria down, all of the bacteria are dead, but the bacterial walls are intact. So whatever stuff they have, it is all present inside. The first step is resuspension of the bacterial pellet using we call it solution 1, 2, and 3. Here the solution 1 is made out of glucose, tris HCl, and EDTA. All of them have different purposes. So as I said, you do not want to rupture the bacterial walls, cell walls yet. You want to let them stay for a while because that will help us to just specifically take out the DNA and leave everything else inside the bacteria. So first you put the solution 1 is made out of first glucose. What glucose does is that it makes a isotonic solution. So when you make an isotonic solution, since the bacteria is not alive anymore, if you put them directly into water, they will just rupture. They will taking a lot of water and this rupture into 5 billion parts, and all of this stuff will come into solution and it will be a mess. You won't be able to just pick out your plasmid DNA out of that. So if when you resuspend, you use a solution of glucose, isotonic solution, which prevents bursting. The cells are intact, but just now in solution instead of a pellet. Next, you use tris HCl. This maintains the pH of the solution to 8, not 8 really, like 7 to 8, so that the cells again do not rupture due to abrupt changes in pH. And finally, EDTA. EDTA, if you have not come across it before, is a chelating agent. A chelating agent is something that takes stuff out from solution. And EDTA can chelate divalent cations such as magnesium, which is very important because most of DNA damaging enzymes have used magnesium as a cofactor. So EDTA prevents DNA damage essentially by chelating out magnesium. So essentially in the first step, you just take the bacteria and make sure they do not rupture and stay in solution. You resuspend the bacteria back into solution from the bacterial pellet. That is step 1. Step 2 is actually alkaline lysis, the name of the entire method. In this, you treat the bacteria with a solution 2 which contains NaOH and SDS, sodium dicyl sulfate, which is a very strong detergent. Both of them together, what they do is that NaOH goes and punctures holes into the bacterial cell wall, and SDS goes in and then denatures everything. So SDS has two parts to it. So it is a detergent. So detergent always have a polar part and a non-polar part. The non-polar part goes and binds to organic parts of proteins, lipids, everything else, and the polar part binds to polar parts of all of these things, essentially causing them to denature. Denaturation means so everything in the cell, as I had said before also, wants its hydrophobic parts to stay together and the hydrophilic parts to look outside. So essentially they fold into a particular structure which is very stable for them, and in that particular structure only they can function. When you denature something, you essentially open them up, you unfold them and make them into long strands. So they are not folded into very beautiful structures. Supercoiled DNA, or if you have ever seen an image of hemoglobin, it like has four different subunits all folded amongst itself. Once you denature, all of them open up and form long strands, and they have the tendency, once they denature, so all the hydrophobic part is now facing towards the hydrophilic water aqueous solution. The moment you do so, all it is extremely unstable for them and they come out of solution. Essentially, they precipitate and form white blobs throughout the solution because they cannot stay in solution any longer. In this step, entire cell gets denatured and everything comes out into solution. Your proteins, your lipids, your plasmids, your DNA, your cell walls, everything. So this is alkaline lysis. So you essentially denature the entire cell and it has come out as a precipitate and will form a thick white blob at the top of the solution. And the third and final step is called renaturation, in which you use potassium acetate and glacial acetic acid. So in the second step, you had raised the pH a lot because you have put in a lot of NaOH. So you made the solution alkaline. In the third step, you do the opposite. You put in acidic stuff to make them come back to a 7 pH, so a neutral pH. In this step, something interesting happens. Everything that can come back, like everything that is very small, which can renature when the pH is fixed back to its normal state, actually renatures. And those small stuff includes our plasmids that can renature, as well as our proteins, which are smaller in nature. So they can come back into solution and then form a supernatant, which is liquid. A liquid supernatant is formed at this step. Interestingly, the chromosomal DNA, since in case of bacteria it is attached to the cell wall, it does cannot renature back fast enough to not to come back into solution. So only your supernatant will contain your plasmid and small proteins and salts from the bacteria, and everything else, the bigger proteins, lipids, cell walls, cell debris, everything will come down into solution. So after the three steps, what you have done is that you have taken your plasmid as well as some small proteins out of the cell into the solution as a liquid. So this is where we are here. So your liquid stuff will contain the plasmid and some smaller proteins, which when you denature and renature are small enough that they can come back into solution. Everything else, chromosomal DNA, the cell debris, everything will be as a pellet contained in the solution. Then you centrifuge again. When you centrifuge again, all of this comes down as a pellet, and your supernatant still contains your plasmid and proteins. And the last step, what you do is that you do a you can do multiple things here. We would normally what we do is that is a phenol chloroform extraction. It's called a phenol chloroform extraction. So you treat the super. So you have taken the supernatant out, put it into a new tube, which contains phenol chloroform and a third thing called isoamyl alcohol, which is ambivalent. So it is polar as well as non-polar part of it. So when you put that in and spin the entire thing again in a centrifuge, what happens is that anything which with any non-polar parts, which are most of the smaller proteins that had come up supernatant in the previous step, they attach to the phenol and chloroform and form a bottom layer. Most of the highly polar stuff, the salts and the plasmids, they form an aqueous layer on top, and then there is a middle layer of isoamyl alcohol which grabs onto any proteins which are ambivalent, which have a polar and a non-polar group. So essentially you have three different layers after this step: the aqueous layer containing our plasmid which are polar, the middle layer which contains ambivalent proteins, and the bottom layer which contains most of the proteins. Yes, Dilip is asking what is the reason behind nucleoid DNA remaining denatured form. So I believe you mean nucleic DNA, but here we do not have nucleic DNA because it is a prokaryotic cell. I understand that you mean genomic DNA, which is the bigger DNA that codes for most of whatever the cell needs. The answer is really in that only. So our genomic DNA is huge, and renaturation in case of DNA means, so you would know that DNA when it is renatured, what renaturation means is that they form their hydrogen bonds back. The ATGC actually the organic nitrogenous base are actually connected to each other as in with hydrogen bonds. That is what forms the double-stranded DNA structure, the phosphate backbones and the ATGC connected to each other as a ladder in the middle. So in case of plasmid is small enough that entire thing gets renatured back very easily. The hydrogen bonds form back. But genomic DNA is huge. So, for example, a plasmid might be 3000 base pairs long. Each base pair means A on this side and a T on this side. A plasmid might be 3000 base pairs long. But genomic DNA might be 30,000 base pairs long or even larger. So it is so huge that all of them have to come back into the correct formation in order to renature. So all the 30,000 bases have to come back together and form that ladder like hydrogen bonding. If that does not happen, what normally happens is that there are parts which cannot renature back in time by the time the entire solution is neutralized, and then they are still unstable and they go down in solution, go down as the pellet. Also, cell wall, the genomic DNA most of the time is connected to the cell walls of the bacteria. So the cell wall obviously is going down. You cannot do anything about it. It is made out of lipid. It is non-polar. It is non-polar. So it that will precipitate. So it takes the entire chromosomal DNA and drags it down along with it. But there is this interesting thing that happens, that during the second step, if I go back, of alkaline lysis, you are always supposed to not shake the tube the to mix the alkaline lysis solution with your solution 1 and the bacterial pellet. The reason is, if you shake the tube, genomic DNA can fragment and break into smaller chunks. These smaller chunks are small enough that when you renature, they get back into solution, and then they will cause impurities. They will appear as impurities in your plasmid isolation, because these smaller chunks can renature back. They are small enough. So you are always asked to just do this, like very slight, slow rotations of the tube to mix the alkaline solution into the solution 1. Do not shake. If you shake, then genomic DNA will break, fragment, and form smaller fragments which are around the same size as the plasmid. So as the plasmid gets back into solution, that will also renature back. So that is why genomic DNA stays denatured. Is that fine, Dilip? Yep. Okay. So you have done the PCI extraction, you have taken another aqueous layer which is just filled out of your plasmids, and finally you then, so your plasmid is present in the solution. So, but how do I get this plasmid out as a pure plasmid? What you do is the precipitation step. Now you add 100% ethanol. You can add ethanol, isopropanol, whatever you like. What it does is that it decreases the solubility or the polarity. The solution becomes less polar when you add ethanol, because obviously water is very polar, ethanol is not that polar. So what it does is that anything that is not very polar, like salts, precipitate out. Our DNA lies in that range, so that it precipitates out. And when you add ethanol, you will see it and then centrifuge very fast, then you see a white pellet forming, which is your plasmid DNA. Actually not. The white pellet is actually salts along with your plasmid DNA. The actual plasmid pellet is invisible. You get a lot of salt, so you get a white pellet. But essentially, that is the step where your DNA precipitates out of the solution. So your tube just has the a pellet, which is DNA, and a lot of solution, which is essentially ethanol. You just throw the ethanol out and then resuspend, and you have your plasmid. So these are the 5 steps of alkaline lysis method to isolate plasmids. Any questions? And then you can use your plasmid for anything you want. You can do a billion different things, which we'll learn throughout the course, that we use plasmids for a billion different things essentially. But yes, any questions on isolation of plasmids? Okay, did anyone understand anything? If it could be that you don't have questions because you've understood very well, or you did not understand anything. Okay, someone put a thumbs up. I imagine everyone has understood everything perfectly. If anyone has any questions, you can still ask. Okay. So, in plasmid isolation, the purpose of alkaline lysis is, the answer is to denature chromosomal DNA while keeping plasmid DNA intact. Even though this is the answer to the question, okay, fine. This is the answer to the question, but not really, as we had seen that the actual point is taking the plasmid DNA out without and denaturing everything else, and it does not specifically denature chromosomal DNA and does not denature plasmid DNA. It denatures both. It does renature. It just renatures plasmid DNA better than chromosomal DNA so that you can extract it. Okay, I think that is fine. Anyone has any questions? I guess not. Okay, blank is used to lyse bacterial cells in plasmid isolation. The options are tris hydrochloride, sodium dodecyl sulfate SDS, ethanol, or agarose. You can put in what you think is the correct option here. Okay, I will give you a moment to try, everyone a moment to try and figure this out. Okay, still have answers coming in. Okay, let us look at it. Anushka says, okay, between all of the questions, we have answers. We have Anushka who says she didn't get the last question. Do you mean this one or the previous one? Like what did you not understand the alkaline lysis method, or do you have any problems with the question itself? Okay, the fourth question. So did you not understand like what this question means, or did you not understand the alkaline lysis method? You can unmute and say out loud if you want, or you can just type it in. Like, what do you want to discuss here? Anushka, especially, yes. Because in the notes, alkaline lysis is used to lyse both plasmid gene and genomic DNA. Yes, absolutely. Yes, that is what I was saying. That even though the answer seems to be to denature, the correct answer is to denature given by the question itself. So this is from NPTEL side. To denature chromosomal DNA while keeping plasmid DNA intact. That is not true. Alkaline lysis uh, denatures both chromosomal and plasmid DNA, but it renatures plasmid DNA back. But you see that none of the other options are even further gone. So alkaline lysis is not binding to spin columns. It does not selectively denature chromosomal and plasmid DNA. It denatures everything. And it does not even say digest RNA molecules. You see RNA molecules as well when you run the final chain. We have to treat the entire thing with RNase, which is an enzyme that eats away at RNA to prevent contamination of RNA. RNA is also a nucleic acid which is around the same size, a bit bigger than the plasmid, but it can do everything that the plasmid can. Ara Aratrika says, I think it should be to selectively renature chromosomal. So it does not renature chromosomal DNA. I would suggest it would be to denature uh, chromosomal DNA and plasmid DNA while renaturing only plasmid DNA back, or selectively renaturing plasmid DNA back, something like that. But I guess the best answer would be to isolate plasmid DNA while denaturing everything else. Like, I agree with what you are trying to say. Like, the answer does not envelop everything that we have been discussing. So, yeah. Okay. Okay, perfect. And we have a billion questions for this. What is used to lyse bacterial cells in plasmid isolation? We have a tricyclochloride, a B, which is SDS, a lot of B's. Trisha says a V, so okay, I don't know. B, C ethanol, C, SDS, C, B, and B. So a bit confusion. So let us look at it. So we just looked at it. So lysis. So there are three different things that we have been discussing. One is lysis, which is the rupturing of the bacterial cell wall. So opening up of the bacteria. Second is denaturation, where you cause the normally functionally folded stuff to become linear and unfolded, so that they, in case of unfavorable condition, they precipitate out of solution. That is denaturation. And finally, we have talked about a lot of precipitation. We have two steps in which we precipitate, which is solution 2, where we do the alkaline lysis here, where we do the alkaline lysis and our cell debris and chromosomal DNA precipitate. And also here will we add alcohol, where our plasmid DNA precipitates. This is not denaturation, this is just precipitation. They are not being unfolded, they are being kicked out of solution because of unfavorable condition, either loss of polarity, like less polar solution due to ethanol, or in case of solution 2, you put in a lot of NaOH and SDS and they are not at all stable enough to stay in solution. But as the question has asked us, what is used to lyse the bacterial cells in plasmid isolation? The answer is SDS. SDS is what lyses the bacterial cells. Everything else is used for different things. Tris HCl is used to maintain the pH of the solution. Ethanol is used to drop the polarity so that our DNA precipitates out. And agarose does not do anything here. Agarose is absolutely rubbish. Okay, is that fine? Any questions from this? Okay. Next, blank is a part of the chromatin where DNA is loosely packed. The options are euchromatin, heterochromatin, centromere, and telomere. I'll give you a second to figure this out. Okay, so we have everyone has answered A, which is euchromatin, and everyone is correct. So the chromatin in case of eukaryotes is present in two different kind of structures. One is called heterochromatin and one is called euchromatin. So heterochromatin are parts of the DNA which are clumped very tightly together, and these parts of the chromatin prevent any transcription factors to get in and start transcribing genes. So these are inactive part of the entire DNA, or genomic DNA rather, where no genes are being transcribed. On the other hand, euchromatin or true chromatin is parts where it is loosely packed, and loosely packed parts, the transcription factors and the entire transcription machinery can sit and start transcribing genes. So these are the active part of the chromatin. Sir, how can we access the problem solving question bank later after the lecture is over? Okay, so one, the lecture is being recorded and will be uploaded to YouTube. So if you want to go through it again, then you can just watch the YouTube video. Two, you will receive a link on the NPTEL page itself where I will be uploading this PPT that we are discussing. So everything here will be uploaded and you can go through them whenever you want. Okay. Okay, so yes, so tightly packed parts of the DNA, which is called heterochromatin, loosely packed euchromatin, and euchromatin is active where transcription is going on, heterochromatin not so much. Can anyone tell me, if you know obviously, what these orange balls represent? The green obviously is the DNA strand. What is this orange things here? Any guesses? Yes, these are histones. Histones are proteins around which the DNA is wrapped, and depending on how which modification is present on the histones, they can be either packed together really tightly or loosely. So acetyl groups, if they are present on the histones, it means these regions are loosely packed and euchromatic and or active. Yep. And if they have methylation marks at specific residues, people have figured out that some methylation marks can result in euchromatin as well, but most of the time methylation marks result in heterochromatin. Perfect. Blank is part of the chromatin where DNA is loosely packed. It is euchromatin. Okay, moving on. So now we are going taking a step in a different direction, talking about plant cells. In chloroplasts, water breaks down into proton and oxygen occurs in which of the following? Photosystem 1, photosystem 2, photosystem 1, ATP synthase, or the lumen. Give me a second to figure this one out. Okay, so we have a lot of questions, answers coming in. Most of them are A, which is photosystem 2. We have one option, which is B, which is photosystem 1. So let us look at it. So obviously chloroplast is the organelle which is specific to the plant cells, and here is where photosynthesis takes place using the light pigment chlorophyll. So the entire thing, the entire machinery for photosynthesis is present in uh, like there are structures inside the chloroplast called thylakoid membranes, which are interwoven membranes, which look something like this. And they have a lumen which is present inside them, and they have the part outside which is present in the thylakoid lumen, so the stroma of the thylakoid. So here we don't have much. We have so yeah, this is what I wanted. So during photosynthesis there are two parts being used. One is called the photosystem 1 and the photosystem 2. The photosystem 2 is the one which absorbs the photon from the sun and breaks down water into protons or hydrogens and then oxygen. And this proton is passed on from the outside or the stroma of the thylakoid into the lumen of it. And here you essentially make a proton gradient. So there are more and more protons accumulating here, less and less coming up here. So essentially there is an electrical force across the membrane, which is then utilized by ATP synthase to form ATP. So essentially photosystem 2 is the one that breaks down water into two parts using the energy from the sun. But when you make a proton, you obviously make an electron as well. This electron is transferred along using the electron transport chain to photosystem 2. This photosystem 1. So photosystem 2 breaks down the water into protons and oxygen and an electron. Protons are used to make ATP. Electron is guided along the electron transport chain to photosystem 1, and the photosystem 1 uses it to make another molecule which is important for the cell, known as NADPH, which is again used in different stuffs throughout the cell. So essentially using photosynthesis, it makes both ATP and NADPH. And the ATP and the NADPH, so obviously if you had learned about photosynthesis ever, you know that the general photosynthetic reaction that we write is 6CO2 plus 6H2O plus solar energy. This maybe sometimes we will write. And then that makes glucose plus oxygen. But the photosystem 1 and 2 together make only ATP and NADPH. Where is the sugar coming from or the CO2 being used? So this ATP and NADPH is simultaneously used up in something known as the Calvin cycle, which uses CO2, starts with CO2 and then uses the ATP and NADPH produced by photosystem 1 and 2 to form actual glucose. So essentially this is the entire process, but it is divided into separate parts. Photosystem 2 breaks the water into protons and electrons. Photosystem ATP synthase uses the protons to make ATP. Photosystem 1 uses the electrons to make NADPH. The thylakoid membrane uses ATP and NADPH and the Calvin cycle to make glucose. So even though this is very simple, but you have a lot of things, complicated things going on in between. So that is how photosynthesis works. So in chloroplast, water breaks down into proton and oxygen occurs in photosystem 2, which is option A. Any questions? Okay, if there are none, we move forward. The next question is, eukaryotic cells secreted out solid materials of the cells through a process called blank. Options are endocytosis, pinocytosis, exocytosis, and phagocytosis. Okay, okay, it seems everyone has answer C, which is exocytosis. So let us look at it. So in the cell, most of things that come in from the outside or are secreted outside do so in single-walled vesicles, which look something like this. These are like essentially balls of membranes that have turned in on itself to form a spherical structure, which are called vesicles, and these go either outside the cell or are taken inside the cell. When the cell takes something inside it, it is known as endocytosis, which endo means inside and cytosis means just engulfing or eating essentially. How that works is that the membrane folds in on itself, forming a cup-like structure, and whatever is remaining inside is then trapped inside this cup as this part closes and forms a vesicle inside the cell. And this then travels to different parts of the Golgi complex where they are treated and things are done to them. There are two parts to endocytosis where first it forms the early endosome which has just come off the endosome, the cell membrane, and then it fuses with the lysosome, which is, we know, the trash bags of the cells. They contain huge hydrolytic enzymes and acidic pHs that destroy everything that is present in the endosome, and then it becomes a late endosome after this fusion, and it travels to the Golgi complex where Golgi essentially takes everything useful out of it and then utilizes it to make more protein, more sugars, more lipids, etc., etc. This is called endocytosis. This can be specific for a particular kind of thing that is getting endocytosed, or it can be non-specific. There are a lot of types of it. A specific type is called pinocytosis, in which the cell essentially drinks liquids from the outside, and they are like very small vesicle which just take in liquid all the time from the outside, which is called pinocytosis. The vesicles are smaller in this case. And finally, there is something called exocytosis, which is secretion. So if a particular uh, protein needs to be secreted out of the cell, say a hormone or an enzyme that acts on extracellular matrix, these all of them are excreted out of the cell. It is known as secreted out of the cell. It is known as exocytosis, where the vesicles start forming in the Golgi and then travel to the outside to the cell membrane, fuse to the cell membrane, and then whatever it was inside is pushed out. So exocytosis. So eukaryotic cells secreted out solid material of the cells through a process called blank endocytosis, pinocytosis, exocytosis, or phagocytosis. The answer is exocytosis. Perfect. And if anyone is interested, you can actually look up phagocytosis, which is everyone is really interested in this thing nowadays. Everyone is doing research, a lot of research on this. Okay, next. Early endosomes fuse with blank to form late endosomes. The options are lysosomes, Golgi complex, plasma membrane, or rough endoplasmic reticulum. Okay, we have answers coming in. Okay, so everyone has answered A. Perfect. So yes, early endosomes do fuse with lysosomes to form late endosomes. You have already seen that. Yeah. Okay. So I'm not discussing it further. But interestingly, a point of interest for you is that some viruses, such as dengue, what it does is that when it is so dengue viruses, obviously you know that they have go into the cell and take over the cells in order to make more viruses. When they get into the cell via endocytosis, they are not active. But when they enter the late endosome, where lysosomes cause their pH to drop because the lysosome has acidic environment, it causes the virus to become active and then starts doing all of more virus preparation. So it essentially has the evolved to use our own systems of defense against us. Okay, so early endosomes fuse with blank to form late endosomes. The answer is lysosome. Okay, the next question again from a lysosome is the lysosome lumen, which is the inside of the lysosome, is blank. Okay, let's look at it. We have one A which is alkaline. Everyone else has answered acidic. Yes, so as I said, lysosomes are acidic in nature and they also contain a lot of hydrolytic enzymes. So enzymes that chop off everything that enters into them and then act as the trash bags of the cell. So essentially, you want anything destroyed and broken down into its usable smallest components, you send them to the lysosome to get degraded. So these are essentially spherical structures with hydrolytic enzymes and a very acidic environment which denatures protein and then chops them up, and it is used for degradation of ingested food material, for delivery to the vesicular system, degradation of pathogenic bacteria, and degradation of old proteins which are not working well. Okay, and this is an image of the lysosome. So we have a matrix of the lysosome, the plasma membrane, and the hydrolytic enzymes. And this is a The lysosome is a double membrane structure, so you have two membranous membrane layers, and anything that fuses into it will just get degraded because of the acidic environment and the hydrolytic enzymes. So the correct option is D, acidic. Okay, next question. We have 15. We have to go till 15. Majority of the plasma membrane is composed of the following. Option A is carbohydrate and nucleic acid, B lipid and protein, C lipid and nucleic acid, and D carbohydrate and protein. Okay, so we have a lot of B options being done, and yes. So let us look at it. So what is the plasma membrane? So I will take this here. So this is an electron micrograph. So essentially you do electron microscopy and you can zoom in to the plasma membrane of bacterial cells, and this is what the plasma membrane looks like right here. And actually it looks something like this. So plasma membrane is a lipid bilayer, which means there are two layers of lipids, and as you know, the lipids have two different parts to them. A hydrophilic head part, which is these balls right here, and the hydrophobic tails, which are right here. So obviously the hydrophilic parts like the water environment present in the cytosol as well as the extracellular space, whereas the hydrophobic tails like each other much more. So the hydrophobic tails point inwards, while the hydrophilic heads point outwards, both into the cytosol and the extracellular matrix. Also, these plasma membrane is home to a lot of proteins, like a huge amount of proteins are present on the plasma membrane. There are two major types. One is known as a peripheral protein, which is present on one side of the plasma membrane. So it is present, like it, some part of it is buried inside the plasma membrane, and the other one side of it is pointing out towards one end. So say this particular one is pointing towards the cytoplasm. Whereas there are other integral proteins which are present throughout. So one part outside, one part inside, and one part towards the cytoplasm. So these have three different parts: the extracellular part, the intracellular part, and the membrane part, the transmembrane part. So these kinds include receptors or channels which connect the outside of the cell to the inside and help in transport of various things through it. So our plasma membrane is impermeable to most things. So it does not let most things inside, especially polar stuff. It does not allow inside. It is impermeable to water, and using these channels, we actually help ions, vitamins, and water to come through into the cell. Without these, no transfer. So essentially, the plasma membrane is 5 to 10 nanometer big, very fragile membrane, but it protects us from most of the stuff outside the cells. It is a lipid bilayer with the polar ends facing outwards, non-polar ends facing inwards. The structure is best defined by the fluid mosaic model, which is something that, essentially, what it describes is that these lipids are not like building blocks that stay where they are. They always move like a liquid, and a lipid here sometime later will move along the entire plasma membrane to somewhere else. So this was known as the fluid mosaic model when it was proposed. It is in a fluid state, yes. And interestingly, so essentially it is made out of majorly lipids. The plasma membrane majorly made out of majorly lipids and proteins, but the top of the plasma membrane also has a lot of sugar molecules, like a lot. The entire cell membrane is covered in sugar molecules, and this is known as the glycoprotein code that is present on top of a cell. Essentially, anything that wants to detect if this cell belongs to the host. So, if our body's immune system wants to figure out if this cell particularly belongs to the host, it first interacts with the glycoprotein coat on the cell membrane, and that acts as a signature that if it is a cell of my own body or it is a invading organism or a pathogen. So our blood groups, the ABC O blood group types that we ABC, AB and O blood group types that we have for the red blood cells, all of them correspond to particular sugars that are present on the red blood cell coat. So these essentially act as signatures and are very important for our body to process if the particular cell is external or belongs to itself. And this is the image which describes the same thing. And yes, the majority of the plasma membrane is composed of lipid and proteins. This is fine. Okay, so we have a question. I have a question. During endocytosis, if engulfed material goes to the lysosome, would it not damage the engulfed material, such as proteins, due to low pH? Yes, that is the idea. So what you want is not functional proteins to enter the cell because that might harm the cell. When the cell engulfs an endocytosis something, the intention most of the time is to break it down to its original component. So that it would be great if the cell can get direct amino acids from the proteins. It does not need the functional protein to come in. It uses the lysosome to first denature and then break it down into a primary structure and then break it down to its original component. So a big protein like hemoglobin, if it comes in, it will be chopped up into different amino acids that it has. If sugars come in, such as sucrose, it will be broken down to glucose and even smaller units. So lysosome helps in degrading them to the smallest parts, which are probably safer for the cell to use directly. It does not endocytose stuff to use, such as like it does not endocytose stuff such as functional proteins or functional nucleic acid, because that is essentially you engulfing a bacteria or engulfing a virus which can take over the cell. So it always tries to damage the protein, but till the level of it still being a building block that can be used somewhere else. So the okay, there's something here. So the role of the lysosome is to convert polymers like proteins to monomers like amino acids. Yes, its major role is degradation and breakdown, but not always to monomers. For in some cases, it breaks them down into tripeptides, which can be directly used. You don't have to waste energy into joining three peptides into like three peptides together to form a tripeptide. So, but essentially, yes, it breaks down polymers into monomers, but majorly it degrades everything that gets there. So that's a very interesting question. Why does the lysosome membrane does not break itself by its hydrolytic enzyme content? So lysosomes contain hydrolytic. So there are different kinds of hydrolytic enzymes. So like trypsin or lipases. These all have specific motifs that they can detect and then break down. They do not break down everything. So this like you would know that our tears has lysozyme which help in the killing any bacteria that are present around our eyes. So these enzymes detect particular motifs. The lysosome membrane does not have any proteins that have these motifs, at least pointing inwards into the lysosome lumen. If it is present, it will be pointing outwards where the hydrolytic enzymes cannot reach them. So whatever present, but most proteins will have these particular motifs that can be detected by the enzymes and then degraded. So something similar happens with our stomach. So we have trypsin being released in our stomach, which degrades everything, proteins into smaller and smaller peptides for consumption and then absorption by the intestines. But why doesn't it harm the stomach itself? Because it is lined against all of these enzymes, and all of these enzymes cannot act on the proteins that are present openly on the stomach lining. Is that fine? Hello, Dilip, is that fine? Does any Does any linings exist on the inner membrane of the lysosome? Not really. It does not have the proteins that are present on the inner membrane. Just do not have the motifs for the hydrolytic enzymes. I was giving an analogy to the stomach, which actually does have a lining. Okay. The various so moving on, the various antibiotics or nutrient supplements should be added to medium when the temperature is blank more than 50 degrees, below 0 degrees, less than 50 degrees, and more than 100 degrees. So for context of the question, this is talking about media that is you are making to grow cells, and normally the media initially contains only water and some things such as agarose and things that are heat resistant, but antibiotics or nutrient supplements, they get charred at higher temperatures, so you add them at a later stage. But what stage do you add them at is the question. And okay, so most of us have answered C, which is less than 50 degrees. Answers are still coming in. I will give you a second. Okay, even more C's. Okay, so yes, we will just have a discussion, but all of you are correct. It is less than 50 degrees. So growth medium. So when we want to grow bacteria, we want to grow mammalian cells, we want to grow yeast in laboratory conditions, we give them food in the form of growth media. So these are liquid media in which the bacteria can grow. These contain stuff that the bacteria can use to make the four major types of things it needs: proteins, carbohydrates, lipids, and DNA or RNA, as well as some minerals and vitamins. So there are different kind of growth media, but essentially they will contain some part kind of nutrient source as well as some part kind of antibiotic to screen for the bacteria that we want to grow. So to prepare, you first take 1 liter of distilled for microbiology purposes. So you growing bacteria. To prepare, you use 1 liter of distilled water, and you then you put all your media components into the water. So essentially mix everything together, and then you then autoclave with for 121 degrees Celsius for 20 minutes, which kills everything that is present inside. So you have sterile media. Then you just pour it down and then put your more and more nutrients in, which can have been charred at higher temperatures, because once they are charred they cannot be consumed by the bacteria. So you do not put glucose directly and then autoclave it, because it will just burn up and then give a darker color and cannot be used by the bacteria. And then once it is cooled to less than 50 degrees Celsius, then you add antibiotics and other nutrient supplements. How do we choose mediums for different cultures, types of cultures like bacterial fungus? Okay, this is a good question. So different bacteria and different fungi, even different mammalian cells, require different kinds of media to be used. Majorly, these have been figured out using people who have been working on microbiology for a long, long time, and they have figured out exactly what is required for bacteria to grow the best in. So, for example, DM-EM media, which is used for mammalian cells, it has been figured out to the T what exactly is required for the bacteria to grow, or rather, the mammalian cells to grow. But again, this most of it is empirical in nature, so people tried different things and they figured out what exactly you need to give the cells in order to grow. There are again two types of medias in this case. One is called synthetic media, in which you know exactly this is the amount of calcium phosphate that I have to put into the media, which will help the bacteria to grow. So these are very specialized in nature and grow only one or two different kind of microorganisms. The other is a compound medias, where you do not know the exact composition. So, for example, LB media, Luria Batoni media, which is used to grow E. coli more throughout the world, like that is the most probably commonly used microbiology media ever. it uses something known as yeast extract. So you ground up yeast and use the entire yeast cells to put into the media, and the bacteria will take whatever it needs out of the solution. So you do not know exact composition, but you know that it works. Similarly, for mammalian cells, you use DMEM plus FBS fetal bovine serum, which and serum has different hormones and different vitamins, which we do not quantify exactly. But these are empirically known that it will help the bacteria or the cells to grow. So these are all have been figured out using like loads of experimentations. But what is the basis of media selection? Like the amount of carbohydrate present for culture to uptake? It depends on the cells. So, for example, for mammalian cells, we know that there are different types of amino acids that are present. Some are called essential amino acids, which the cell cannot make by itself. So it needs to be supplemented from outside the different things such as valine, phenylalanine, alanine, tryptophan. All of these are essential amino acids because the cell cannot make it by itself. So if you want to make a media for mammalian cells, you have to put all of these in the media. But non-essential amino acids can be made from glucose itself. So you put in a lot of glucose and the cell will use whatever it wants. Same for tryptone. Tryptone is amino acid supplement which is then converted into different nitrogenous sources that goes into DNA and RNA preparation. But again, all of this has been majorly experimentally figured out that this is the exact amount of stuff that we need, and then people calculated stuff and then figured it out. How do cells take up glucose from DMEM? If that okay, so how cells take up glucose from DMEM if cells are not present? So when you put in the serum, the FBS, serum is the part of the blood which is devoid of red blood cells. It is supposed to have everything, every hormone that the cells need to grow. That then it also contains insulin and growth factors which help the cells to grow. You do not put in insulin specifically, you put in the serum entirely and it would contain stuff that the cells need to grow. Bacteria don't care about that, but mammalian cells do, and that is why you specifically have to put in serum. There are different experiments in which you do not put in serum, which are known as serum starvation conditions, and we see that if only DMAM is present, even though there is glucose around, the cell starts to show signs of starvation. So all of the serum is important because the hormones help it to uptake. Is there any non-insulin dependent glucose uptake process? There is. There it is known as GLP glucose like insulin glucose like insulin GLIP mediated GLP or GLIP mediated glucose uptake, but that is secondary method. Like major for mammalian cells is going to be insulin. Okay, the various antibiotics or nutrient supplements should be added, and it is obviously less than 50 degrees Celsius. Next, plants and bacterial cells are more resistant towards osmotic lysis due to presence of what? The options are thick cell wall, thin plasma membrane, vacuoles filled with water, and the presence of villi or flagella. Mm, what's the need of compound medium? Okay, what's the need of compound medium where there is already exists a defined medium? Yeah, so you need a compound medium because it is first easier to make. You do not have to measure out exactly the amounts of hormones, vitamins, nutrients that need to go in. And secondly, compound media, defined media are used in specialized conditions such as virus making in particular kind of cell lines where you need to know exactly what is going in so that you do not have any kind of contaminations, and you know these kind of cells grow definitely in this. But in case of compound media, it is not that specialized. You can use it for most cells in most conditions, and it is obviously cheaper than a defined medium. I think that is fine. Okay, coming back to this question. Okay, then yeah. Plant and bacterial cells are more resistant towards osmotic lysis due to what? And we have the answers coming in. We have a C and a lot of A's. Sorry, the C has been changed to a A. Okay, fine. And then the A. And okay, so everyone says cell wall. That is absolutely correct. So how this works is that whenever, since the osmotic pressure inside around the cell causes water to go in the cell, if it is only protected by a cell membrane, then as we have seen, it is a very thin membrane and very fragile. Water goes in, it just ruptures the cell. But if it has a cell wall, which is in case of bacteria made out of peptidoglycan, in case of plant cells made out of cellulose, it is very solid, and even as the cell expand, it cannot expand as much and does not rupture herein. So it is like a balloon which is you have covered it with your hands from all ends. You just can't inflate it because your hands are covering it. So it never ruptures. So on that note, we will just look at the differences between plant cells and animal cells. So cell wall, yes, cell wall is present in plant cells and it is made out of cellulose. Animal cells, it is mostly absent. Size, plant cells are larger, animal cells are pretty small. Chlorophyll is present, animal cells is absent. Vacuole, plant cells have large vacuoles, which are these organelles which just store food and water and stuff. And animal cells have a small and many in number, so smaller vacuoles. Mitochondria few because they depend mostly on lysosomes. Animal cells have much more animal mitochondria. Lysosomes almost absent, animal cells much more, glycoxysomes present, animal cells have them, do not have them, cytokinesis by plate division breaks in the middle and forms higher cell walls at the same time. By construction, just the cell membrane pinches off, and chloroplast, obviously, plant cells have them and they do not. Can you please show the last question again? Okay, do you mean this one or do you mean this one? 13th. Yes, I think I have placed the 13th question after this because the two were one after the other. So yeah, we will see the 13th after we are done with the 14th. We are not like yes, the next one is the 13th. Yeah. Okay, the plant and bacterial cells are more resistant towards osmotic lysis due to presence of a thick cell wall. Now the 13th question. In mammalian cells culture media preparation media cannot be autoclaved as it might degrade the media components. For that, the media should be blank. So since in mammalian cultures we use a lot of nutrients and serum, if you heat them up, they will all degrade. So use something else. What is it that you use? They are heated to 100 degrees for 10 minutes. They are filtered using sterilized membrane filters with a pore size of 0.22 microns. Heated to 100 degrees Celsius for 10 minutes and sterilized using ultraviolet light. Okay, so everyone is answering B. A and C and the same. Yes, good point. That drops it from 25% to 33% that we will answer correctly, but everyone has answered it correctly. It is B, filtered using a sterilized membrane filter with a pore size of 0.22 microns. So let us look at mammalian cell culture. How do you do it? So almost everywhere the mammalian cell culture we use DMEM, which is this DMEM actually is predefined what exactly goes into it, and this is in the form of powder. You mix it with sodium bicarbonate and put it into water, and that gives you a reddish color to mammalian cell culture medium. So if you have ever seen cells being grown in a cell culture room, they always have a red colored appearance in the plate. Then you add FBS and then your antibiotic, and then filter the entire thing using a 0.22 filtration 0.22 filter. But can anyone say why is this red in color? Does anyone know? No, I don't think so. Phenol red, that is actually correct. DMEM actually has a dye called phenol red, uh, which is indicator for. So what does the red color mean? And like why does it need to be red? So what does it tell you that it is? What for is it red for? Well, like, what is it red for? Like how does it indicate something? Okay, so I will answer this. So phenol red. So what it does is that it is a component of DMEM, and in the physiological condition, which is slightly acidic, it is red in color. So you always see red colored when the cells are growing very well. But when the cells start dying, what happens is that where you grow your cells, you always grow your cells in a 5% CO2 condition, which balances the pH of the media. The moment the cells start dying, the entire media turns orangish in color, which says the media needs to be either changed or all the cells have died. So it indicates that you have a very well functioning uh, cell culture, or otherwise if it turns anything other than this red, then you can just throw the plate away because everything has died already. Yes, it is a pH indicator as has been pointed out. So in the media cannot be autoclaved because it might degrade media components, and that is why they should be filtered with a membrane filter with a pore size of 0.22 micrometers. Can anyone tell me that what is the 0.22 filtration for? What are you filtering? Bacteria. Yes, but that why do you need a 0.22 micrometers filter? Why not larger? Okay, I will just tell you that 0.22 is does filter bacteria, but also filters viruses. The mammalian cell cultures always have this problem of virus infections coming in. So 0.22 filtration prevents viral infection as well because that prevents viruses to come through. That is why I use the 0.22 micrometers. If you do not care about viruses, you can use a zero point 0.4 micrometers filter as well, which is majorly used outside cell culture because most bacterial cells do not care about viruses. Like we do not care that the bacterial cell is may be infected with viruses. But with mammalian cells, they die very easily. So you want a 0.22 micrometers filter. Very good. Next, in blank, physical cell disruption methods. Cell disruption habit happens via cavitation bubbles. The options are thermolysis, osmotic shock, sonication, and pestle and motor. Okay, so we have a lot of some of these people. Okay, so it is indeed sonication. So in sonication, you use this in order to rupture cells, but not very harshly. What you do is that there is this probe, so this entire mechanism, and here at the tip there you attach a probe. This probe looks something like this. So it is like a tuning fork, and you put your cells inside a falcon or something similar, a stand, and put the probe inside the tube. and then you turn on the sonicator and vibrates at a very high frequency. Vibrating at a very high frequency causes cavitation bubbles to form, which are essentially air bubbles that are pushed out. Not air bubbles, like these are water bubbles made out of water that are being pushed very fast towards the walls because it is shaking so fast. So essentially, it creates so much vibration inside the tube that it breaks apart of the cells. This is how sonication works. So it generates ultrasound frequencies at 20 kilohertz, causing cell disruption by cavitation, causes compression and decompression rapidly, causes gives shock waves to cell wall plasma membrane to cause cell lysis, and okay, essentially you break the cell wall apart using vibrations. So the answer is sonication for this one. Okay, that's all I had for you today. Where you can see these questions after the session. Yes, that is what I said. I will upload the entire PPT, uh, and it will be available to you on the NPTEL homepage for molecular biology. So if you just go to the molecular biology page, you will see a section saying problem solving sessions, and once I start uploading the PPTs, you can see them up there. And also this entire session is being recorded, so the entire thing will be on YouTube, so you can go ahead and look at the recording later as well. Yeah. The next session will be on the second week's lecture. Absolutely, the next Saturday we will have the second week's lecture sessions. Okay. Okay, if that is all, thank you everyone for joining. I will see you all next week. Bye bye.